Hello and welcome. This is the Nutritionist webinar. I am Marianne Fessenden from AMTS. Today we are joined by Dr. Peter Yoder, Technical Services Manager at Perdue Agribusiness. Peter obtained a BS in Animal Sciences from the Pennsylvania State University, apparently that's a thing now, in 2010, and an MS in Animal Sciences from the Ohio State University in 2012. That's been a thing for quite a while. And a PhD from Dairy Science from Virginia Tech in 2018, with a focus on dietary nutrient variation and amino acid metabolism. Peter's industry work experiences range from internships farms in California and Nebraska, as well as being employed at Perdue Agribusiness since 2013. Peter grew up on a small dairy farm in central Pennsylvania and enjoys spending time there. He also enjoys international travel when he can, learning about cultures and volunteer work. It's wonderful to have Peter join us today. Remember, while the presentation is pre-recorded, Peter will be joining us in the end for questions. Please now enjoy Dr. Yoder's presentation. Remember to jot down any questions you have during the presentation and type them in the Q&A or chat windows. We will have an opportunity for him to answer those questions after the presentation.
Good morning or good afternoon. I appreciate the opportunity to present here. Thanks to the AMTS team and Marianne for this opportunity. Today I'm going to talk a little bit about my PhD work and then some associated research. But the primary focus of the talk is going to be on amino acid metabolism, especially post-absorptive, what's new, and at the end, some ideas that we could possibly translate to field nutrition. So again, thanks for the opportunity, and I hope this topic is somewhat useful to you. So big picture, and this is looking at primarily the U.S. dairy industry, but I would say this probably translates on a global basis. Typical dairy cows have about 25% efficiency of converting feed nitrogen or feed protein into milk protein. So approximately 75% is going to end up going into the environment, either through urea or urine or the feces. So from an efficiency standpoint, could we change that 25% to 30% or 35%, and therefore reduce environmental nitrogen load into the environment, which again would make our industry much more sustainable. So one of the reasons I think this sustainability is somewhat important, and this is more focused here in the U.S., 13 counties produce 25% of the milk. This is as of 2012, so I'm sure it's more concentrated today than 2012, but we have larger and larger farms, highly concentrated, very small geographic area, leading to significant ammonia emissions into the air, and also nitrates into the groundwater. To the right here is the state of Idaho. As you can see, the red dots there is where nitrate levels exceeded the drinking water standard recommended. As you can see, where major dairy counties are, we see a lot of red dots. So this efficiency going from 25% to 35%, given the consolidation we're seeing in the industry, is going to become even more critical going forward if we want to have sustainability for the environment. Also, when we look at amino acids and protein, I think, and this is going to go into formulation, we see changes in ingredient prices and we see changes in protein costs. Here on the left is blood meal. That could be soybean meal. We could put canola meal, but the prices change and quite dramatically. This is looking for the last 12 months here. As you can see, blood meal just in the last month has decreased almost 40 to 50% in price. Should that change how we formulate diets? We do have least cost optimizers in AMTS and other software that helps address this. So from an ingredient standpoint, we can manage that somewhat. But when I look here at the right, this is metabolizable protein, the cost estimated coming from sesame. And as you can see, over time, that's changing as well. So if the price of a nutrient such as energy or metabolizable protein changes, should I change how much I formulate to in a diet, or should I follow what the strict requirement is? And this is something that came about, I guess, through the beginning of my PhD and led to wanting to study post-absorptive metabolism. Is when the price of a nutrient changes or the price of the output changes, should I change the target formulation? Here's an example of the output. This is milk protein. This is in the US, but on a global basis, I would surmise that milk protein changes in price as well. And as you can see here, you go back 2-3 years ago, milk protein was around $1.20 to $1.50. Today, it's approaching $3 to $4. So if milk protein doubles in price or it declines by 50%, should I change how I formulate a diet? Should I change how much protein I feed? Should I change how much of specific amino acids I feed? We look at it from a sustainability standpoint. When the price of milk protein is low, say $1.20, $1.50, perhaps I should feed a low protein diet, maximize sustainability as well. So here's an example, and this is straight out of AMTS. This is an example diet I put together here. As you can see, the total supply of MP is 2,836 grams. The requirement is slightly less, so we have an excess of 60 grams. So the question, and you can see MP milk is 92.2 pounds. So if the price of MP changes, price of metabolizable protein per unit of pound changes, or the price of milk protein changes, should I change what I formulate for? When the price of milk protein is low, should I formulate for less MP milk? And when the price of milk protein is high, should I formulate for more? Somewhat looking at this as a response function. As I feed more and more protein, I do get slightly more milk protein. As I feed less MP, I get less. I think in animal nutrition and using models, particularly on the MP side, we tend to think of requirements and not a response surface. For the energy side, if I overconsume calories, if I consume more than what my requirement is, there is a response. I put on body weight. I think we see that in dairy cows as well. So moving from a requirement basis versus a response basis, I think is critical, and I'll get into why we need to look at it from a post-absorptive standpoint. So this is the first part of my PhD work, is we look at formulating diets, and I think traditionally we would look at ingredient costs. What's the price of soybean meal, price of corn, price of blood meal, and the nutrient requirements. We'd say, as an example, we need 90 pounds of milk from MP prediction, or 2,900 grams of MP, and we would lease cost to achieve maximum income over feed costs. But if we add in the additional variables of milk component prices, such as the price of milk protein, a nutrient response surface. So if we believe we feed more protein or we underfeed protein, something happens to the animal. And we add in nutrient costs, so the price of that metabolizable protein or the price of that amino acid, such as methionine, as well as ingredient costs, we may lead to a different solution. And I put that together here in this publication, as you can see here below if you have more interest. But I think this is where we need to go here on the right. The problem is, from a post-absorptive metabolism standpoint, we struggle to predict nutrient responses, which I'll discuss here in a bit. And so once we have a model that is predicting nutrient responses and looking at milk component prices, we can start looking at marginal economics. So when the milk price on your client's farm doubles, should I change how much methionine, or should I change how much MP I formulate for? You probably should, but our models don't help us achieve that, and that's what I'm proposing here on the right-hand side. So that was first an overview of sort of why I wanted to study post-absorptive metabolism, to predict marginal responses, to better understand how to manipulate efficiency. What I'm going to go through right now is some more background of where we're at, and then get into my research studies. But protein supplementation today, using CNCPS, I think does a very good job at predicting supply. We can predict grams of lysine, we can predict grams of metabolizable protein fairly well. Of course, there's some opportunity for improvement, such as better understanding microbial protozoa flows to the duodenum. But we've definitely made progress using computer software such as AMTS to predict supplies. These models, whether it's AMTS, CNCPS, or NRC, predicting milk protein or predicting allowable milk, simply multiply times a coefficient of 0.67. So if I have 3,000 grams of metabolizable protein supply, after I subtract off maintenance needs and needs for growth, I simply multiply times 0.67 and I get the allowable milk. This doesn't take into account energy supply. It doesn't take into account amino acid profile. And it doesn't take into account where am I at on that curve? Am I oversupplied or undersupplied, and what is the marginal efficiency? So in some ways, post-absorptive metabolism for amino acids or protein is really an inflexible black box. I can add methionine to a diet. I could feed less protein. I could feed more energy. And the model still predicts the same output. So we really need to, I guess, have a better understanding of regulation and predict in order to better predict milk production and milk protein. So here's an example. This review article was published about 2 years ago by Dr. Helen LaPierre. And she took a previous study from Wisconsin out of Dr. Glenn Broderick's lab. You can see there's 5 levels of crude protein here. And on my y-axis, I have milk protein yield, grams per day. And what you can see here, this black line is observed. So this is what the cows actually did in that study. The orange line would be CNCPS or AMTS. And the blue bar dotted line is NRC. So you can see at a low protein diet, CNCPS did very well, 15%. NRC still missing by a lot. CNCPS a little better. And you can see as we increase protein, both models, whether you're NRC or CNCPS, are over predicting output. The other trend I'd like to point out is notice how each model, whether it's CNCPS or NRC, is sort of a linear increasing function, and not necessarily a plateau or smaller rate of change as we observed here in the cows. So really, 13.5% protein diet, 1,100 grams of milk protein, 16.5% crude protein diet. We get close to 1,200s, and then we start to flatten off in cow responses. But our models are predicting this either significantly under predicting here or over predicting here on the right hand side with the high crude protein diet. And this relates to that 0.67, so that fixed MP efficiency. It's not accounting for improved efficiency when I feed less protein, and when I overfeed protein, it's not accounting for reduced efficiency. This study here is a little bit dated. It was a meta-analysis by Dr. Dopal, and as you can see, it's illustrating somewhat what we saw here, the problem in the previous slide. As I increase metabolizable protein supply, my efficiency goes down for converting it to milk protein. So let's say around 1,700 or 1,800 grams per day of MP, the efficiency would be around 40%, the marginal. As I increase out to 2,500, close to 3,000 grams, that efficiency is declining down to 0.2. Our models, whether it's CNCPS or NRC, are using a 67% efficiency. So you can see as we overfeed protein, the model is going to overpredict the responses. When we underfeed protein, the model is actually going to underpredict what's going to occur. This has consequences when we look at changing dietary formulation. Let's say soybean meal or canola meal or rapeseed meal is very expensive, and I want to feed less MP. If I'm using CNCPS or NRC, I feed 100 grams less MP at a 3.1% true milk protein, it's going to predict a 5-pound loss of milk, or about 2 kilogram loss of milk here. However, if I use a more reasonable marginal efficiency, instead of using 67%, I would use 20 to 25%. Going to predict only about a pound to a pound and a half loss in milk, or about a half a kilo to 0.7 kilograms of milk. So then the whole economics of that change, or that of proposing whether I feed less soybean meal and thinking I lose 2 kilograms of milk when I may only lose half a kilogram of milk. So I hope that somewhat makes sense of how using that fixed efficiency, when we underfeed protein, the consequence is probably much less than what the model would say. The same if we oversupply. So if we believe in strict requirements, overfeeding protein, overfeeding MP above the model requirement should yield no extra milk. If you believe the response surface of the model, it'll actually suggest significantly more MP milk. Reality, you probably have a declining efficiency. You are going to see somewhat of a response, and it may be half a kilogram to 1.3 pounds of milk. So again, the economics, I think, of changing supply using our current model structure can really mislead us on the economics of that decision. The other, I guess, reason to study post-absorption metabolism is the incorporation of other factors influencing milk protein yield, such as energy. This is from that same paper, and what they're looking at is a previous study done at Virginia Tech by Dr. Mark Hanigan and Dr. Alex Rios. And as you can see here, high energy, this is low energy, low protein, high energy, low protein, low energy, high protein, and then high energy, high protein. And this is milk protein yield here in the y-axis. What you can see is the cows respond to energy in milk protein output. The models, particularly the NRC, is not predicting much of a change, especially here in the high protein, same. And CNCPS is somewhat of a change, but not nearly as change as we're experiencing in the cows. So I think this points to that 67% efficiency. We probably need to adjust that. When we're feeding a higher energy diet versus a lower energy diet, the conversion of MP or amino acids in the milk protein is going to change, and it's going to be higher. So accounting for that in formulation may help us better optimize milk protein output. So again, you can see here, NRC, high protein is way over predicting responses because energy is a limitation. So to summarize here, this part one here, optimal nutrient supplementation for income over feed costs likely varies. So as the price of milk changes, you may want to change your dietary formulation. As the cost of MP changes, cost of supplying protein changes, we may want to change formulation. However, your marginal response predictions by models are likely inaccurate. And that goes back to that black box of 67% efficiency. When the marginal efficiency is probably more like 20%, 25%, using 67% efficiency is going to lead to a very different calculation of marginal income over feed cost changes. So again, better understanding description of post-absorption metabolism is likely needed to address that. Here's an example, and this would be US based in May 2020. This is the price of milk protein. This is the price of metabolizable protein. This is the price of energy. And this is right in the midst of the COVID-19 crisis here in the US, so many farmers were discussing feeding less, reducing feed costs, reducing production. If I feed less NP to save some money, the models are going to predict this marginal return of minus 42 cents. However, if I feed less NP here and I'm using a marginal NP efficiency of 19%, the marginal return might actually be break even or slightly positive. And the same for overfeeding protein. So again, I think it points towards we need a better understanding of post-absorption metabolism so that we can make more informed decisions when economics dictate. So moving to part two, I'm going to talk a little bit about how we looked in my research, a better understanding regulation post-absorptive. This is just a simple graphic here. So we have absorbed metabolizable protein arriving at the gut and liver, and there is catabolism there, and some of that is going to be excreted into the environment through the form of urine. It also has a flow to the udder, flow to the other tissues, and then also flow back to the rumen and large intestine. So you look and account for that loss of nitrogen, approximately 24%. Remember, our 25% efficiency is going to go out in milk protein. 57% is going to go out into the environment in this simple, from this paper here. So when we look at this, is this black box, what is causing the regulation of uptake by the udder, uptake by other tissues, catabolism by the gut and liver, and then excretion into the urea into the environment. Much has been done in this area, but I think there's still more to do. We understand that group 1 versus group 2 amino acids, catabolism by the liver versus catabolism in the udder, and transformation of non-essential amino acids. There are distinct differences there, but what could we do to further, I guess, stimulate milk protein synthesis so that this 24% is more like 35% or 40%? And what is our understanding of amino acid transport and uptake by the udder was two areas that I specifically looked at in my research. So amino acid balancing traditionally, I think this is how we've probably all been trained to a certain extent, is looking at the barrel and stave and what is the most limiting amino acid relative to our output milk protein. In the US, it's thought to be methionine and lysine. So if methionine is the most limiting amino acid, then that's how full this barrel can be of water, or that's where our milk protein output is capped. This is very inflexible when we look at this. It does not account for a change in efficiency of use. So what if my absorbed methionine to what's taken up by the udder, what if that changes? It does not account for energy. What if I stimulate more milk protein through energy, which I'll get to? Those factors don't go into it. And of course, when you look at a meta-analysis put together by Dr. Hanigan a number of years ago, it poorly predicted responses. And then when we look at adoption, particularly here in the US, you'd estimate maybe 15 to 25% of cows are actually fed according to this. So the vast majority of cows are not adopting this. So is this completely explaining post-absorptive metabolism and regulation of milk protein, or is there other things at play? The other thing is, if you believe methionine is the most limiting amino acid with the barrel and stave, if I increase that methionine supply, basically bring up the water that can be stored in that barrel, the efficiency should be close to 100%. If I count for maintenance and catabolism by the liver and those tissues, I could bring that down to 50 or 60%. But when you look at the data, and this is a meta-analysis by Dr. Van Amberg, the marginal efficiency is only around 11%. If I take this same chart out of NRC 2001, I believe they found 10% marginal efficiency of methionine to milk protein methionine. So if it's truly a limiting amino acid in the barrel and stave, I think we would agree that number should be at least 50 to 60%, if not closer to 100% efficiency. Being at 11%, am I oversupplying it, or is there another function that's causing this stimulation? And I challenge you, look at it in one of your everyday herds that you're working with. For example, if I have 80 pounds of milk, or let's say around 35 kilograms of milk, and I change milk protein concentration from a 305 to a 315, that's about 36 grams of milk protein. And I think many of us who work in the field have done that change, going from a 3.05% to 3.15% through methionine supplementation. So 36 grams of milk protein, that's 1.1 gram of milk protein methionine. How did you get to that? You probably supplemented a rumen protected methionine, probably around 10 to 12 grams of MP methionine. You change the diet, well, that's 10% marginal efficiency. So I would challenge you to go back to some previous examples where you implemented amino acid balancing and look at the marginal efficiency, and I think you'll find it's much less than you would expect. Moving forward, and this was work done by Dr. Ryan Higgs and the Cornell group, and I think it makes a whole lot of sense, was to tie digestible methionine or MP methionine to energy. So we'll get into energy somewhat regulates milk protein, and so having that relationship, I think, makes a lot of sense. However, one of the questions I have when we look at this is, we're seeing this 1.1 or 1.15 is the requirement. Well, you can look here at the data and you can see, yes, at this 1.15, this might be where the first derivative changes. The slope's changing, but we can have cows out here at 0.9, 1.0, and have higher methionine efficiency. We can be above that and have lower efficiency. So really, it then becomes an economic question. What is the price of milk protein? What is the price of milk in general? And where do I want to be on that efficiency of use from a marginal economic standpoint? I think too often we think this is a requirement when you can actually see from this data it's a response, and this can mean significant changes in economics, whether I'm at a 0.9 here or 1.15. The other thing to think about, I think this 57% efficiency, this is where that derivative is changing to get to that 1.15. You look at group 2 amino acids such as leucine, isoleucine in the CNCPS model, their efficiency of use is higher. That doesn't, from a biological standpoint, and there's group 2 amino acids being used to make non-essentials, their efficiency should probably be lower than methionine, which is a group 1 amino acid. So some other questions, and this is just a simple study I just wanted to point out, again, looking at this barrel and stave approach. This is done by Dr. Bill Weiss at Ohio State, and they had a control, and this was primarily corn protein. Then they supplemented methionine and lysine, and then this treatment 2 which I'll get into a little bit what that was. But you can see crude protein's the same, MP's the same, MP methionine changes, of course, and then your lysine to methionine ratio changes. And these are estimates out of NRC. I think we would all say this diet here is probably going to do the best, right? If we look at the barrel and stay, we would say this diet is going to outperform because we're supplying methionine. Well, the cow said otherwise, and as you can see here, milk increased the most for treatment 2, energy-corrected milk as well, milk protein yield, which again, we're increasing 40 grams of milk protein yield over a treatment where methionine and lysine were supplemented. You can see that here in the arterial methionine concentrations. So how did treatment 2 happen? Methionine should have been the lowest stave, and I still got a greater increase in milk protein. So again, I think that's pointing to other points of regulation. Branched chain amino acids in the arterial were much higher here. So what I'm not telling you here is this control was basically all corn protein. It was a corn silage diet, and then they fed a corn byproduct as a grain with it. So no soybean meal, no canola meal, no alfalfa protein here. They added methionine and lysine in this treatment, and then treatment 2 would be our more traditional diet where we'd have soybean meal, heat-treated soy, and alfalfa protein. So the takeaway from this is I think we focus so much on methionine and lysine. Well, what about these other amino acids, and is there other points of regulation? So another study, I guess, pointing to this, this concept of looking at methionine and lysine that I think should raise some flags was this study out of UC Davis by Dr. Peter Robinson and Dr. Swanepoel. And they took, I think it's 20 commercial dairy farms, average production around 45 kilograms. They looked at the high cow pens of those dairies, and they measured the blood plasma lysine and methionine. As you can see here on the right, the ratio in the blood plasma really did not, it wasn't related to milk production. You had ratios anywhere from 2.25 to over 4, and there's no relationship to production. Look over here on the left-hand side, here's methionine concentration, lysine concentration, especially some variation there on the lysine side. Again, no relationship. The blood plasma concentrations are what the mammary gland is going to see. This is showing me that whatever the ratio was in the blood plasma, there doesn't seem to be a good relationship with milk production. So is there something else at play here? I did a cell culture work, and this was my first study based somewhat on that previous slide. What we did was we wanted to say at the mammary gland level with epithelial cells harvested from a cow, if I change the lysine and methionine ratio, if I change the profile of all amino acids, how does that affect responses? So I took this blood here would be blood meal, casein, of course the profile in casein, corn, profile in corn, cow, what's the typical profile of amino acids we see in cow, and DMEM, just a standard cell culture treatment of amino acids that's used. As you can see, the ratio, big differences here in the lysine methionine ratio, big differences as well in certain amino acids such as isoleucine and lysine. Again, corn being very low in lysine, blood meal very high. Well, what did the cells say? I looked at 4 different proteins that are associated with making milk protein: mTOR, S6, 4-BP1, EIF2 alpha. Really no difference. So whether I balanced for lysine and methionine or I did not balance, there was not a change in phosphorylation state. So I guess it begs the question of, does the profile really matter at the cell level or not? I do think the profile matters to a certain extent from a substrate standpoint, but the cell has some flexibility for managing the imprecision that may arrive at the udder. And that led to amino acid transport work. And I think it can be very basic, but I think it can really change your mindset when you look at amino acid nutrition in a dairy cow. This over here on the left-hand side is an in vitro model that we put together looking at leucine. This is measured results from cell culture. And as you can see here, this extracellular, which would be outside the cell, intracellular, and then the protein turnover pools, catabolism, and then also transfer into keto acid. What you can see is 72% of the leucine is being effluxed. So you have leucine coming in, and then leucine going right back out. So very rapid bidirectional exchange. And this is not new. I mean, this has been observed in amino acid transport for a number of years, in that there's a large number of transporters. Some transporters are unidirectional, some are bidirectional, and there's different points of regulation. So what I'm not showing you is here is the full results from the trial, but as intracellular amino acid concentrations change, efflux is going to change, and to a certain extent, the influx will change. So if I'm low on methionine in my diet, there may be less efflux, which then will allow the cell to maintain intracellular amino acids. And I think this is one way where imprecision in our daily supply. So we have our diet, the amino acid profile varies. At the cell level, intracellular, we need precision for translation, but transport can change the profile to meet the needs of the cell. Just here on the right-hand side, this is pool turnover, just showing you how rapidly that entire pool can change over. Leucine, for example, is turning over within 7 minutes. So very rapid turnover between the different pools to match what's needed by the cell. Then we went and looked at this in vivo, say, does this actually happen in a lactating dairy cow? These are lactating dairy cows producing 45 to 50 kilograms of milk. We used stable isotopes, and we looked at all amino acids. I'm just showing you two groups here. So it'd be isoleucine and leucine. And what you can see here is significant efflux going on. 40% of it's being effluxed. Methionine and lysine, somewhat the same story. So somewhat matched up with what I showed you earlier in vitro, we are seeing that rapid bidirectional exchange occurring in the udder of lactating dairy cows. So if methionine is limiting, this efflux can decrease. The same when methionine is overfed, this efflux is going to increase. So there's ability for the cell to use transport to manage the precision to translate RNA into protein, which you can see here on the left. So take a typical dairy cow, and let's say we're feeding her 62 grams of methionine. If this is the efficiency of use from absorption to milk protein, we're going to predict a negative balance of 24 grams at 45 kilograms of milk and 3:1 true protein. If her efficiency is 90%, if she has blood flows high, if there's strong stimulation intracellular, if transport is reducing the efflux, you're actually going to have a positive balance. And I think this range here, 57 to 90%, is definitely possible. If you go back to that slide I showed you from Dr. Higgs and energy, you can see that range in efficiency occurring. So just to end this section on transport, if you have further interest in it, I would recommend you look at these two reviews. This one here on the left by Christensen, he's sort of the pioneer in amino acid transport, and I found that paper to be an excellent paper for starting to think about transport as a method for regulating homeostasis of the intracellular amino acid profile. The idea of thinking of the diet profile being what the cell sees and not having flexibility is likely not true at all. And amino acid transport plays a big role in managing the variation in diet to have a very homeostasis environment in the cell. Same here with on the right. This is a very good review looking at that as well. So moving from, I guess, cell transport to looking at inside the cell, what regulates milk protein or anabolic activity? Again, we've talked about the barrel and stave a lot, but are there other points of regulation? One example I like to give is you think about de novo milk fat. So de novo milk fat, for a long time, we thought it was related to substrate. Is it related to acetate, fiber digestion, etc.? Not until late 1990s, early 2000s, we discovered CLA. And CLA's ultra-biohydrogenation and effect on fatty acid synthase, and basically the machinery that makes milk fat, de novo milk fat in the mammary gland. Significant impact. I think for milk protein, we're learning there is that function as well. Can we change the enzyme activity, the machinery that makes milk protein in lactating dairy cows? This paper that I have up here above, I would recommend checking out. The mode of, I guess, regulation of anabolic activity or protein synthesis across a wide range of species is thought to be mTOR, and you can see that here at the top. We look at just lifting weights or trying to put on muscle. I mean, what do people do? They take branched chain amino acids, they take whey protein, they take leucine. You're not really talking about limiting amino acids. You're talking about how do I stimulate greater protein accretion. Here on the right-hand side, so does that translate to a cow? And I think there's been a lot of debate in this area. The research is a challenge to do because there's so many points of regulation of growth or anabolic activity. We first say there are studies out there showing that it may not be linked in dairy cows. It may not be linked in other ruminant species, but I think it's worth exploring. So just summarizing it real quickly. So this is mTOR, looking at hormones, amino acids, and energy. Can that change the machinery that makes milk protein? We've looked at it in vitro, this paper right here, and seen responses. We've looked at it in rats and seen responses. And in dairy cows, especially in short-term trials, I think this was one of the first studies to show that this mode of action is affecting milk protein synthesis. So really, I guess a crude way to look at it is, can I change the machinery? Can I speed up the engine or stimulate milk protein using some of these factors? From an amino acid standpoint, leucine definitely just seems to be the most potent. Methionine is as well as several other amino acids such as arginine. Just another picture of looking at that. Here's amino acids here on the left-hand side. We traditionally think of the barrel and stave and what's limiting to going on that tRNA when we have all these other points of regulation, whether it be mRNA abundance, ribosomes, the abundance of those ribosomes. We have energy going into to translate that protein. Even out here, we look at the formation of the protein. There's some thought that endoplasmic reticulum or unfolding of proteins plays a role into it. So the saying is there's many other points of regulation here. When you just look at the barrel and stave, I think we're missing these other points of regulation. In vivo evidence, this would be greater than 4-day trials. This study, Virginia Tech several years ago, saw no response to supplementing branched chain amino acids. This study, also Virginia Tech, saw no response to supplementing leucine. And then this study at Guelph, again, saw no response supplementing branched chain amino acids. So you may say at this point, well, our theory that translates to dairy cows is probably not true, seeing these 3 studies. And they did not find in looking at mTOR or there's other proteins associated to it, they did not find increased phosphorylation or increased activity. So is there other is this theory not translate to dairy cows, or are there other factors that are at play that are blocking activation? And that's somewhat what led to me thinking there's other things at play here. So if you go through all these studies that looked at leucine or trying to achieve that mTOR response, very low milk fat or very low production. You can see these two studies, 2.5 milk fat, 3.1 milk fat, 2.8 milk fat, or also very low production for a Holstein in early lactation. So when I have low milk fat, I do have endoplasmic reticulum stress. Is there negative feedback blocking stimulation of milk protein or anabolic activity? And I would argue there are some links from a protein signaling cascade that can be made that would block activation. So to put that in layman terms, if the cow's sort of messed up from a metabolism production standpoint, why is she going to increase anabolic activity? And I think that's part of the reason in these previous trials we did not see responses. So in a cow study that I did here several years ago, I said, I want to ask that question again, but I want to make sure the cows are healthy and producing very high levels of milk. In this case, they were producing close to 50 kilograms of milk. Supplemented, infused 2 groups of amino acids, your traditional methionine, lysine, histidine, or group 2, which would be more the mTOR related amino acids. We would say there should be no responses to group 2. Treatments, just real briefly here, it's a 2x2 factorial. So we had our control, then we had methionine, lysine, histidine, isoleucine, and leucine, and then the combination. How did I come up with these amounts? Well, this first MKH was based on AMTS model, CNCPS, to put it out there, methionine, lysine, according to grams per megacal of energy, and histidine, just looking at it relative to methionine. Isoleucine and leucine, that was sort of a best guess estimate based off what we see when we feed high RUP diets. So here again, just looking at it in a model, you can see our methionine and lysine, where they would be recommended by the model, and then in our control and isoleucine, of course, they're low. So what did the cows say? Here on the left-hand side, this is milk protein percent, and you would say, oh, we're right, methionine, lysine, histidine, the barrel and stave amino acids, I got that nice response in milk protein. Leucine and isoleucine, no response. Numerically, a negative response. So maybe these amino acids aren't playing a role. But we look at pounds or production, we did see an increase. And I'll show you next slide. It was driven through milk volume. So here we're seeing independent increases of milk protein from 2 separate groups of amino acids. And in combination, we're seeing the greatest increase here in milk protein. So in this gray bar, the IL, isoleucine and leucine, how did it increase milk protein independent of supplying methionine when we know methionine is deficient because the cows responded to it? We'll get into that here in several slides, but I think it's pointing to there are other points of regulation or stimulation that can be used to optimize milk protein. Point out here in the yellow bar, you can see this additive response where we're getting some response from methionine, lysine, histidine, some response from isoleucine and leucine, and together get the greatest response. Milk production, so here's the gray, that would be the mTOR stimulating amino acids, isoleucine and leucine. We see a response, about 4 pounds or 2 kilograms of milk, and energy corrected milk as well. So are we stimulating greater anabolic activity, which is leading then to greater output? I'm not going to go into all the, I guess, description of blood arterial amino acids or net uptake by the udder, but I would encourage you to check out the publication. We saw significant changes in the blood plasma, blood flow, net uptakes out of the udder that helped explain why we saw the responses that we did. But briefly here, just looking at methionine, and this is venous. So just as a reminder, venous is the blood exiting the udder. What you can see here is when we supplement methionine, lysine, histidine, it's nearly 4 times higher. You can also see that versus IL. So much of the methionine that we added actually exited the udder. So what is that methionine going to end up for other tissues, or is it just going to be catabolized by the liver? You can see the effects on marginal methionine efficiency. So this is how much of the methionine we fed or infused ended up in milk protein. You can see in our MKH, only 9%. That's very close to the 11% we talked about earlier. We add in isoleucine and leucine, we could double that. So we stimulated milk protein, milk production by some other essential amino acids, and we're now at 20% use of that methionine. This is very different than this red bar here. So that's the overall efficiency in the model, 57%. So as I feed more and more methionine, declining efficiencies, and when I look at the marginal efficiency of methionine or any amino acid, it's probably much less than what we would infer from the model. And here is just looking at overall efficiency of use. Here's methionine approaching 90% when we don't supplement it, which begs the question, is methionine for other functions in the body, is it being limited? Potentially yes, but we do supplement it. Significant drop down here in efficiency into the mid-60s. Same for lysine, same for leucine. So this red line is what the efficiency is in the CNCPS model, and you can see, especially for methionine and lysine, the cows could probably perform okay, though they do get a stimulatory response or respond when we add it, but that marginal efficiency again would be much less than what we'd infer. So this whole thing on leucine and isoleucine, I showed you several trials where they saw negative or no responses. We discussed how maybe low milk fat and metabolism not being optimal limited those responses. I showed you my trial. That was a very small study, my trial, so I always question whether my trial actually is real and can be translated to the field. Here's several other trials. This is one trial recently published in JDS. They had a negative control, methionine, threonine, and then they added isoleucine and leucine, and they saw a tendency over methionine to get a response. And for milk yield, milk volume, they saw a response. Lower producing cows here, but did see positive responses from isoleucine and leucine. A meta-analysis also recently published associated leucine with increased milk and milk protein yields. I would say, just looking at abstracts from this last year at ADSA, there are some abstracts that point to positive responses as well. So talked a lot about stimulation, mTOR, leucine, isoleucine. Does methionine and lysine still matter? Yeah, they probably do, because there is still a substrate requirement, but I think there's variation in that efficiency that can change it to being not only substrate, but also stimulation of milk protein. Here's a trial recently published as well. You can see milk protein grams per day. This ILV is isoleucine, leucine, and valine. Group 1 is what GR1 stands for, so this is going to have methionine in it. You can see it gives a greater response than just supplementing the branched chain amino acids. So probably needed here. When we look over here at fat protein corrected milk, again see a greater response when we supplement the branched chains versus ALT, which is arginine, lysine, and threonine. So is there that stimulation response? And I think this trial would confirm it, but also suggests, we can't just completely forget about substrate requirements. So to summarize this section, or part 2, I guess here in one slide from my PhD work, is don't think about a cow and amino acid nutrition as being inflexible. It's not a machine. It's not like an airplane where if I have a shortage of one fuel or something changes, that we have an issue. The cow has tremendous flexibility to survive. It's part of the evolutionary process. She can handle significant variation in amino acid profile through blood flow, changes in blood flow, through changes in amino acid transport, through changes in signaling at the cell level. And I think that changes the way we formulate. We think of, if I'm short 5 grams of methionine, that's a big issue. Well, it may be an issue, but think about there's varying efficiency. Maybe if I give 5 grams more methionine, it's more stimulatory response because having 5 gram deficiency, she's surviving just fine because she's using it with higher efficiency. So start to think about not just amino acid requirements, but a response surface and a variable efficiency, amino acid transport playing a role in this flexibility. And then this last point, I really think finding that unique optimal essential amino acid profile likely doesn't exist. There's so much that changes from absorption at the gut to when it reaches the udder and transport that various amino acid profiles can likely achieve the same thing. Though I would agree at some point, if methionine or lysine is too deficient, there would be issues, but there's flexibility in metabolism to handle various profiles. So part 3, just moving to some field nutrition ideas, application. Predicting milk protein yield, this is a meta-analysis by Dr. Mark Hanigan, likely will be in the next NRC. But these are the factors, this is the regression model for predicting milk protein yield. Digestible energy, body weight description of the cow, digestible NDF, and then there's 7 essential amino acids. We're not just looking at methionine and lysine. And when I look at this, I put 3 stars beside leucine and methionine. We have good evidence in vitro and looking at other experiments in other species. There is a mechanism for stimulating mTOR, being stimulatory amino acids. Same holds true for arginine and isoleucine, though the connection is not quite as strong, and the Km or the concentration intracellular that leads to stimulation is not as close to in vivo as leucine and methionine. So then you have lysine, that's probably more a substrate-driven response. Threonine, there is some potential related to stimulating mTOR, and then histidine, probably substrate. So looking at 7 essential amino acids instead of just looking at lysine and methionine, and I would challenge you to think about which amino acids stimulate, which amino acids probably more a substrate requirement. So when you formulate diets, again, I think this is somewhat, you probably all do this today, but maximize microbial protein through fermentable carbohydrates, starch, that's going to bring some of that digestible energy. It's also going to bring the hormones. It's going to bring the insulin response that we're looking for to further stimulate as well. Multiple essential amino acids. I'm going to get into this on the next slide. AA efficiency varies. So when I'm formulating a diet, think about the marginal economics with more reasonable efficiencies, and then consider this regulation. Could I use this to be more flexible in formulation? So this slide, and I just put this together, this is more theoretical. This is not necessarily, I guess, I don't have hard data to back this up, but it's something to think about. Is this is income over the cost of supplying methionine supplementation to cows. So here in the y-axis would be the income over the cost of the investment. And I have 3 different marginal efficiencies of using that methionine. So 10%, 20%, and 30%. And then I have 2 different prices of milk protein. I think when you just supplement methionine and energy is not optimized in the diet, and I had lower other essential amino acids, and I'm post-peak, I'm probably going to get 10% efficiency. And that's what the NRC The data set that Dr. Van Amberg put together, that's what they showed, 10%, 11% efficiency. If I'm just looking at milk protein, I probably shouldn't supplement methionine because of the negative economic responses. However, if I use energy, I use other essential amino acids, I try to stimulate responses, I have the interaction of energy and methionine more appropriate, I'm probably going to get a higher efficiency. And if I can approach 20% efficiency, I get much more favorable economics. So here in the high milk protein, I'm close to 25 cents a cal a day. This last one, all stars align. It's sort of just thinking about, is it peak lactation? I have my fermentable carbohydrates such as starch, sugar optimized, doing very well there. I have other essential amino acids such as leucine, histidine, isoleucine at levels. Then when I come in and supplement methionine, I may approach this 30% efficiency number. Then we get these very significant responses. So I think this is a different way of looking at it than just that barrel and stave where I only look at methionine. What if I look at these other factors, and if I can optimize them in synchrony, I'm going to increase probability of a positive return on my investment. So some strategies to consider, I would look at targeting what I classify mTOR amino acids plus your limited essential amino acids. This is a traditional approach of just look at methionine and lysine. I would propose in diet formulation, either look at total essentials or look at methionine plus lysine plus leucine. In particular, I put stars between isoleucine and arginine because I think we have less confidence in their stimulation of mTOR, but definitely on leucine, I think there's strong mechanistic data showing responses there. So when I'm changing, I guess I want to optimize milk protein, I could either feed a lot of methionine and lysine and get my targeted response and have a low efficiency, or maybe I spend the same money, feed less methionine and lysine, but feed some of these other essentials and get a similar response. I would encourage you to check out this abstract from JDS. It was just last week, and they showed somewhat looking at this approach of having your traditional amino acids and your mTOR amino acids, and they see different responses and stimulation when they change energy. So just putting some economics behind that, when I look at methionine by itself, metabolizable methionine per pound is very expensive. Synthetic is usually the most economical go-to, and you can see that with $11 a pound. I look at soybean, canola, or blood meal, very little methionine brought in a metabolizable basis. If I add methionine, lysine, histidine, which I'd categorize as your traditional limiting amino acids, you can see how these ingredients sort out. But what if I look at mTOR essential amino acids? And here, this calculation, I'm looking at methionine and leucine. If I look at methionine and leucine from mTOR standpoint, you can see these feed ingredients are very favorable. Heat-treated soybean meal or blood meal. And then lastly, just looking at the gross total essentials. So when devising, I guess, diet formulation or thinking how to maybe apply some of this to the field, I would suggest, why not target 20% efficiency or 30% efficiency, which means I can feed less of this expense of the highly priced methionine and use other ingredients to drive mTOR essential amino acids and get the same response. Or I could try to get even greater response and feed increased amounts of both. One other question, I get this a lot in the field, and I think there's some more research going into this, especially with the Cornell group, and I'd just like to comment on it a little bit. But we look at methionine, lysine, or essentials relative to energy, which I agree is probably the best approach. But does energy from fat versus energy from glucose propionate change our recommendations or how we think about it? And I would argue it probably does. So here's two. This is a paper. This is an abstract. Both of these studies, they looked at fat and they looked at glucose, and then they changed amino acid supplementation within those two scenarios. Fat gave independent responses, independent of amino acids. Glucose does not. And so I think when we're driving up energy with starch or digestible fiber or something fermentable carbohydrates, I definitely think we need to raise our amino acids. When we drive up energy with fat, I think we still need some more studies on it, but my initial hypothesis is that we probably don't need to increase supplementation because it seems to be an independent response. And it probably goes to insulin responses. So when I have greater propionate, have greater insulin, greater anabolic activity at the tissue level, I need more amino acids, and fat does not elicit that response. So in summary, to wrap this up, substrate versus stimulatory essential amino acids, I would challenge you to think about that. Don't think the barrel stave is a strict regulation of milk protein. There's other factors that play a role. Energy can stimulate, mTOR through stimulation of certain essential amino acids like leucine can stimulate it. So it brings this question of strict amino acid requirements. I think it's going to be really hard for us to identify because of the flexibility, that change in efficiency. So whether I have greater stimulation or less stimulation changes probably where I should target amino acid supplementation. Secondly, consider marginal responses to MP amino acids. So it's probably much less than what the model would say, both from MP and an individual amino acid. So how can I increase that and optimize it and get a better return on my investment? Being at 10% marginal efficiency for methionine, what if I feed less methionine but drive up energy, drive up other amino acids such as leucine and histidine? Does that give me a better response, better economics? And then I would suggest looking at that. And then when we look at balancing, supplementing for amino acids, again, I would look beyond methionine and lysine. In my opinion, when I'm working in the field, I track total essential amino acids first and then consider the individual amino acids and realize there is flexibility. So just to wrap this all up, I think our models do a very good job predicting supplies. When you take supply to output, there is definitely some work I think needs to go into predicting output and changes in marginal supply. So I think we still need to use our intelligence and cal responses and think about, here's what we're seeing in research, here's what we're seeing as metabolic flexibility. How could I manipulate that to optimize income over feed costs, farm economics, but also looking at this and efficiency, sustainability. If protein is very expensive, if milk protein is less, maybe I just feed less protein and optimize inefficiency, and vice versa. So it's just definitely thinking outside the box. When you see requirements, I would put uncertainty around those requirements and realize there's flexibility and this response level regulation. So with that, I'll take questions, and I'd like to acknowledge Dr. Mark Hanigan, who is my advisor. He did contribute significantly to this work, and I appreciate his mentorship. And I'd also like to thank the funding, which was Perdue Agribusiness.
First of all, again, thank you, Peter, for the presentation. Let me introduce you to our next month's speaker, who is Jennifer Van Os. She's an assistant professor and extension specialist in animal welfare on the faculty of the Department of Dairy Science at the University of Wisconsin-Madison. Dr. Van Os received her PhD in the Interdisciplinary Animal Behavior Graduate Program at the University of California, Davis, and conducted postdoctoral research in the Animal Welfare Program at the University of British Columbia. The research in her lab at UW-Madison focuses on understanding, evaluating, and improving the welfare, the welfare of dairy animals from a biological perspective. Next month, Jennifer will talk about managing heat stress to improve the welfare on dairy cows and calves. So join us for that presentation, followed by a live question and answer session twice on August 13th. The presentation, as with this one, starts at 9 a.m. Eastern Daylight Time in the morning or 6 p.m. Eastern Daylight Time. We run it twice. So as you know, the webinars take a lot of work and cooperation. They are organized and produced by AMTS, USA and Global. In the afternoon, we are joined by Paula Turillo of Athena, who hosts this series as El Webinar del Nutritionista. She receives support from Guillermo Lerman, Tecnal Rock River Lab in Argentina, Biotur, and Concar. She has the translation skills of Paula Alanes backing her up. We also thank AMTS distributors in Italy, Elena Bonfante of Dairy Innovations Italia, who is with us this morning. And she also is joined by her partner, Bill Prokop of Dairy Innovations. They will both be on as panelists. From China, we have Sean Lee of Ansci Tech. He will join us this afternoon. And from Brazil, Marcelo Hens Ramos, director of 3R Lab, and he will also join us this afternoon. Marcos Neves Piera of Universidade Federal de Lavras in Brazil is in the afternoon session. Marcos makes a point every month of sending thoughtful questions in the months he's unable to join, so we're happy to have him here with us this afternoon. We are very grateful for our sponsors who make it possible to get wonderful speakers and manage the program. We thank our gold sponsors, Arm & Hammer Animal Health, makers of cattle feed ingredients that optimize dairy cow health, and the Canola Council of Canada. Learn more about feeding canola at canolamazing.com. Our silver sponsors are Ajinomoto Heartland, superior nutrition through amino acids, makers of AgiPro L, Dairyland Laboratory, and Virtus, makers of Strata with EPA, DHA, omega-3s, and Prequel with omega-6s. Our bronze sponsors are Dairy One Forage Laboratory, Amino Max, Adiseo, Perdue Agribusiness, PMI, and Soychlor. Each of these companies support education and research worldwide. We hope that you will consider them in your formulation decisions. I'm now going to open up the floor for questions. Again, for the English language listeners, I will read your questions for you. Remember to type them in the Q&A tab or the chat window. Elena will be asking questions from the Italian perspective. And we also have the noted Tom Taluki and, as I said, William Prokop joining us this morning as panelists. So I think y'all know how to open up your own mics and say good morning. Peter, if you want to start your video, you can. None of us usually do. So it's up to you. And again, Peter, thank you for joining us.
Okay. Yeah.
There you are. Hi, Peter.
Hey.
Hi, Bill. I see you're unmuted. Everybody else is staying in silent mode.
Okay.
Good morning. I am here.
Thank you. Thank you. Peter, I'm going to start out with some questions, and then Elena, I'll swing over to you, and Bill, I'm hoping you'll jump in and provide some. We love this when we've got some back and forth. So, and Tom, again, you know how this goes. So my first question is for amino acid and MP research studies, is it worth measuring plasma amino acids? The assay is expensive, and based on what you showed, it wasn't related to much. And Peter, again, if you want me to move to a slide, just tell me.
Okay. Thanks for the opportunity to present, and thanks for the interest as well. Regarding that question, it is expensive, I think, on a commercial setting. I think the variable cost at a lab it can be much less depending what process you're using. If you're using mass spec, I think the cost can be much less. But yes, plasma amino acids, I don't think is a great indication of whether your amino acid supplementation is optimal. To count for blood flow, tissue uptake, catabolism by the liver, catabolism in the mammary gland. So yes, I would say no.
on measuring it. What I would say is, though, I think I have some interest or a hypothesis about measuring venous blood concentrations. I think venous blood concentrations can give you somewhat an indicator of what the blood flow leaving the mammary gland and concentrations there, just sort of as a indicator. But looking at absolute, making inferences on just blood concentrations, even if it's venous, I think you're missing a lot, since you're not accounting for blood flow or tissue uptake.
Okay, all right. Thank you so much. Next question is, can we use MUN as a monitor of protein efficiency?
Thanks for the question. In general, absolutely yes. I look at that. I think we often look at that frequently. I think it's a function of am I optimizing protein usage in the rumen, and then post-absorptive MP, yes. I guess more related to my talk, I would challenge you, if you are in the field and you have good data as far as intake, production, look at nitrogen efficiency, the calculation there, and maybe even consider looking at individual amino acids. You have a predicted supply. Now you're not going to account for scurf or fecal losses, microbial losses, but you could calculate what's your methionine efficiency on a gross sort of a crude level, and see where that comes out. Is it 80%? Is it 50%? I think that could give you some inference on where you're at, and if you do supplement, how that would change. So I don't know if that answered the questions, but MUN, yes, I think it's definitely useful to look at. I think if you want to drill down a little bit, I think there are some opportunities to look at it a little more closer.
Before I move to the next question, Bill or Tom or Elena, do you have anything to add to that? I don't want to step over a question while we might have some discussion.
Well, I was going to make a comment. It dovetails into that somewhat. First of all, very exciting information, Peter. Enjoyed the talk. And your perspective on models, all models, forces me to quote George E.P. Box, that all models are wrong, but some are useful. And I think that that's where we're at very much today. But I think part of the problem, and as a nutritionist, I do a lot of troubleshooting for other nutritionists in the field. And so I see the model used, or perhaps the expectation is misinterpreted in terms of what can we expect to get out of it. And, you know, as you pointed out in the LaPierre study, the model will be predictive if we look at it in total, in terms of energy and protein. If we start to uncouple one from the other, we get disparities, as we saw in the LaPierre data, which was Broderick's rations. My point being, the energy part of this equation that factors into how all of these proteins are going to be utilized efficiently or otherwise, I think are so critical. And, you know, as you pointed out, when we look at the effects, we can't just look at what the MP prediction is. The model is telling us what's first limiting. It's not telling us how it's going to be partitioned. And so we have to look at it from the standpoint of what's our energy corrected effect? What's the total body effect of all these interventions? And to the MUN, I think it's a great tool. The problem we have is lab integrity with the data. And so maybe PUNs might be an alternative. But I am very excited about this, because what we're approaching here is the systems approach at your molecular level to how nutrients are utilized to the carbon, and what the current models are doing on a micro level in the rumen and a macro level in terms of managing the environment. And so it brings a lot of things into play for consideration. So I applaud that.
Great. Thank you, Bill. I'm going to ask one more question on from I have on my side, and then we'll go to Elena. My question is, how can we implement adding essential amino acids other than lysine and methionine in a practical and commercial situation on the dairy cattle farm?
Hey, again, thanks for the question. It's a very good question. From encapsulation standpoint and feeding a synthetic amino acid, I believe there's some test products being looked at, but as far as histidine, leucine, isoleucine, I'm not aware of any commercial products. So when we look at adding other essential amino acids beyond methionine and lysine, we're likely going to do it through feed ingredients. And my comments would be is look at, first, can I improve my microbial protein? Can I optimize the rumen better? And then secondly, do I need to feed more protein? And when I'm looking at protein sources, looking at their essential amino acid content, corn protein versus canola versus soy versus blood. I think there's some ingredients out there that you can simply feed more protein, and you will drive up essential amino acid supply in tandem with methionine and lysine.
Tom or Bill, do you get out there and you see more of what's going on maybe globally? What do you have any thoughts to add for this?
Tom, I'll defer to you.
Well, it gets really challenging because in the majority of the world, animal proteins aren't allowed to be fed. So it's and then even the availability of good bypass sources gets to be a real challenge. Not to mention that the variability in the quality of some of those products. So it's I'm going to go back with Bill and be like, especially if we go back to some of the work from Bauman's group from the 90s and 80s on using euglycemic clamps, that sure, if we want to really fine tune how we maximize milk protein, we've got to be looking at individual amino acids, and that is definitely the direction that we're going. But first and foremost, we're limited by how much energy and what kind of levels of intake we can get into these cows. So I have a real, I guess I can say I've got somewhat of a pretty big concern that we're talking only about how we can manipulate amino acids and to try and have this discussion in the absence of discussing energy, especially knowing what increased glucose supply does to the mammary gland, that we're really missing the largest proportion of impact that we can have on farm. And that's getting as much energy into these girls as we possibly can.
I would agree. I agree. And that, I guess from a research standpoint, we have to be a little bit careful, because a lot of my discussion was just about amino acids, and as we manipulate energy, everything changes. I do think, though, if energy is constant, changing amino acid supplies, I think we change efficiency. If we change energy, we change efficiency of amino acids, in my opinion. But I definitely agree on the goal of trying to get as much intake of energy and delivery of that to the mammary gland.
All right, thank you. Yeah, I think it comes back to the fundamental thing that it's a cow, it's a biological system. And sometimes when we try to turn it into equations and chemicals, that's where maybe it goes off the rails a little. Elena, how are you? Would you like to ask some questions?
Hi, Marianne. Thank you very much. Can you hear me?
Yes, we can, Elena.
Yes.
How the hell are you?
Thank you. Just making sure. Hi,
Ciao,
Peter.
buongiorno.
Buongiorno. Thank you for your presentation. And my question is on the Italian perspective. If we can, that here, for example, in our diet, we use a lot of soybean meal, for example. So, based upon your experience, do you think in those kind of before to consider, lysine as a limiting factor, we should focus on others like leucine, for example, or others?
Okay, I guess I'll repeat your question and make sure I understand it. You're saying that you use primarily soybean meal, and you're wondering if you should focus on other amino acids beyond lysine when you're looking at diets. Is that the question?
Exactly.
Okay. I think it's a good question. I would say yes. I think methionine is definitely probably the first one I'd go to. So when I look at methionine, I think from a requirement basis, from a substrate basis, I think our diets oftentimes are slightly limiting. And I think methionine also stimulates milk protein. There's mechanistic work showing that, the mechanism's there. So I would definitely take a look at methionine in those types of diets. And when you look across all feed ingredients, usually the synthetic rumen protected source is the most cost effective for getting bypass. So in your situation, I would look at methionine and think about methionine having two roles essentially for milk protein: stimulatory and being a limiting substrate. Secondly, if you are, I assume you're using CNCPS, I would look at energy, look at essential amino acids, look at lysine, look at methionine, look at it relative to energy. And I would always try to make sure my total essential amino acid supply is adequate. What that number should be, I think you personally, I probably need to do some more research there. Do look at some numbers in the field, but always trying to make sure if I am going to implement to try to drive milk protein, that my MP, essential amino acid supply, is not going down too much. It's going up. And again, trying to make sure I have a good amount of energy. So don't know if that answers your question or not, but I would definitely look at methionine because there's two roles, and I would make sure my total essentials is the supply is going up. So.
Okay. Thank you, Elena. Do you have any more questions?
Another one. Just refer to the last one of the last slides where you were talking about the energy supply and comparing, the carbohydrate and fat supplementation. But which is the effect in the positive effect, of fat supplementation on trying to increase the MP.
Do you know which slide that is? I can move to it.
Oh, it's one of the last one. Or if you can explain which is the role of those kind of energy sources in MP.
Yeah, I think I'm trying to remember which slide too, Marianne. I think it was.
Well done. This will make it easier.
So I guess first off, the fat versus carbohydrates, and then linking amino acids to energy. I definitely think we should link amino acids to energy. The debate, I guess, our group gets it here in the field is should I feed more amino acids when I supplement fat? I believe, and this is, I put two references in there. If you're using a fat supplement that does not change insulin or partitioning of nutrients, I believe it's somewhat an independent effect. So you can get greater butterfat yield without changing amino acid supply. Now, if you're using a fat fatty acids such as oleic or palmitic primarily, and we are seeing changes in insulin, whether it be insulin resistance or promoting insulin. I think linking amino acids there might make a little more sense. Carbohydrates definitely, because we are going to see insulin changes there. So that's sort of how I look at that. So I think if a fat is just simply not going to have an insulin response, then I don't think you need to link amino acids to it. If it is, I think you do. In your case, I would point you to particularly that Nichols reference. I think driving up essential amino acids, promoting anabolic activity in the mammary gland, that paper, as well as several other papers, show responses to de novo milk fat. So increasing de novo milk fat from amino acid supplementation. So I guess from applied high level view, can you look at, in your situation with wanting butter fat, can I use amino acids to increase de novo, use fat supplementation to increase preformed, and therefore raise butterfat pretty significantly? So I don't know if that answers your question, but that's sort of my comments to that.
Thank you
Okay,
very
and
much.
I had a comment from Jim Aldrich that said fat may alter mammary blood flow from Kant and De Peters.
That's a great point. I remember that paper. I should go back and review it. I definitely agree. It can alter blood flow, and that's going to change delivery of amino acids, delivery of substrates in general, and affect production. So I agree, and I need to go back and revisit that paper. Do you remember what type of fat he was supplementing when it altered blood flow? I'm trying to remember that paper.
Yeah, we'll see if Jim responds. He's pretty on top of things like that. So, Bill or Tom, are you familiar with that paper? Here we go. He thinks it's sort of a review. I'm not sure what that means.
Maybe it was a meta-analysis review.
Okay.
Looking at
Review
fat.
of other studies. Yeah.
Okay.
Sounds like it.
Okay.
All right. I don't have any more questions.
I've got a couple more.
Great.
So Peter, when we look at a lot of these methods that result in increased milk protein yield, be it increasing energy, be it increasing other amino acids beyond, especially methionine, and even in your data it showed we see these increases in milk yield, which in normal circumstances is one thing to talk about. But what we've been going through in the industry recently, and it's not just the US, it's other countries as well, where we don't want that additional milk volume. We want to maximize milk protein yield, but we don't want to, a lot of places are seeing a negative on milk price for excess production. Basically, we go more down the path of processor initiated, might as well say quotas. Would that change your whole approach on how to evaluate some of these responses?
I'll give you my opinion. It's not necessarily based on, I guess, academic research. It's sort of what I've been saying in the field, and absolutely yes. I think when you look at, say, histidine or leucine or some of these responses in the literature where you just feed more essential amino acids or feed more MP, it seems agree with you completely. It raises lactose, it raises milk volume, milk protein yield goes up, but milk concentration does not change. And in today's world, for a lot of dairy farms, that would be a negative financial outcome. So in that case, I'd say, yeah, I think we should probably not implement some of these strategies if it's going to lead to greater milk volume, greater milk lactose through stimulation of anabolic activity in the mammary gland. In that case, then I would divert only to methionine, and using methionine to change the concentration of milk protein, and not, I don't think we would, in my opinion, lysine or some of the other essential amino acids, I would put less focus on them if my goal is only to go from a 3.1 to a 3.2 milk protein and not change volume. So yeah, if your goal is not more volume, then you probably, this thought of stimulating milk protein, stimulating anabolic activity is probably not something, it might not be your optimal solution to pursue. What can I ask you what you think about it?
Oh, well, yeah, no, that's, well, I have a few different ways that I look at it. It really depends upon what is the discount that people are being forced to take on any overproduction.
Mm-hmm.
That really comes into where are we on that marginal return?
Mm-hmm. Mm-hmm.
And then it gets tricky as to try to maximize that component yield by modulating milk volume, which is really tricky.
I agree, and I don't have a good answer to it, but I do think when you supplement these other essentials or just feed more MP, I think you get milk volume, and that could be a negative, and so it is really tricky. It's a different paradigm for sure. It's basically how much milk protein can you get relative to lactose, and it's trying to get that as high as you can get it. I guess is a challenge to do without changing lactose yield.
Well, especially when we start looking at the potential of, especially whey proteins being another osmotic regulator with milk volume, mm-hmm. there's biological limits as to how far we can go.
Mm-hmm. Mm-hmm. Yeah.
The other one is, and this is a more of a theoretical one. Is milk protein really the right end product for us to be measuring against? Because when we start looking at all of the data that's come out in the last few years on the impact of supplemental amino acids, excuse me, on immune function, on repro, to me it seems as if we only focus on milk protein, we could be making some really bad decisions that are where milk protein might be maximized, but we are sacrificing overall animal health.
I completely agree that that's a whole nother area that if we're going to look at it from a systems approach, we must look beyond milk protein as far as health and reproduction. My caveat to that is I think transition cows, fresh cows prior to peak, I think they're we see those responses from amino acids other than as far as reproduction, health, inflammation, etc. I think when you get post peak, and you could argue like some of the studies I discussed today are post peak. I do think at times when you look at just a low overall efficiency we have in dairy cows, 25% on a gross nitrogen. When you look at post-absorptive, getting maybe 40, 45 of use of the MP, and then you look at the marginal responses being at 20%, amino acids being quite low, those marginal responses. I think we need to look at it definitely post-peak when we're not constrained by intake. And I think when you see venous concentrations of methionine change that much, I think the liver is catabolizing a good amount of that extra methionine we're supplementing, especially post-peak. Prior to peak, I definitely agree factoring in what are the needs for the animal in terms of amino acids that we don't compromise health needs to be considered. So I guess that my take on it. I agree it needs to be more than milk protein, but I think once you get past peak, I think we could start to drill in a little bit more in milk protein.
Tom and Peter and Bill. Elena has left us. She had to leave, but it was great to have her join us. I have a question, more of a maybe question comment. This is, what's the effect of amino acids on cell proliferation, apoptosis? That's what should be guiding rationing decisions, although mTOR probably links short-term effects with long-term effects. This is the commenter said he found Kant et al. 2018 Journal of Dairy Science to be really insightful on this.
I found that paper to be really insightful as well. I think that still is the debate whether mTOR is only short-term versus long-term. And I am starting to lean more towards just short-term, and then we have a sort of a longer-term effect once the cell adjusts. But proliferation apoptosis, that is one factor in regulating milk protein yield. If you go back, I think it's slide, Marianne, if you want to go to slide, I'm trying to find the slide 28, number of cells, I mean, that's going to be one of the factors regulating milk protein. Trying to remember specifically that review paper from Dr. Kant, whether there were effects. My hunch is, my memory at this point is that there's not a lot of experimental evidence showing that, but I do think transition cows, fresh cows, it might be something we want to look at. Are certain amino acids or amino acid supply affecting the number of cells, proliferation, therefore changing milk yield for the lactation? So I don't know if that answers your question, but I think looking at slide 28, actually it's somewhat based on that review paper that you're mentioning there, but there are other points of regulation beyond just the substrate, and cell proliferation apoptosis is definitely one of those points.
Okay, terrific. Does anybody have any other questions, comments, things to throw in? I don't have anything more in my windows. All right. So again, Peter, thank you for joining us this morning. Tom and Bill, always a pleasure. We will be back this afternoon, or whatever it makes it for wherever you are. It'll be 6 p.m. Eastern Daylight Time. I know some people joined us a little bit late, so if they want to catch the beginning of it, and I'm sure we'll have a lot of questions coming this afternoon. We have the South American continent joining us from the sounds of it. Let's get started with some questions. I want to welcome Bill and Paula. Marcos is here, but he's going to have me ask questions. Peter, hi.
Hello. Good evening.
Good evening. And Sean Lee's here too. So Paula, before I get started, or Bill, actually Bill had a fantastic statement this morning that I think really put this into some perspective. And it was a quote from George E.P. Box. Bill, would you like to lead us off?
Sure. And good evening, Peter. You have to endure me twice in one day. I apologize. Okay.
It's always a pleasure.
You summed up. I really liked your statement this morning, summing up the complexity of it.
So for the benefit of the home audience, I'll repeat the perspective that I think all of us as nutritionists and those involved in dairy management have to appreciate. And Marianne alluded to a quote by George E.P. Box, the statistician, Nobel laureate, and he said, and I quote, "All models are wrong, but some are useful." And that's exactly what we're seeing today, and probably will always see with models, because we'll never have all the information we need to make them right all the time. But what we have to appreciate here is what we're doing is a systems approach of trying to bring together the molecular system that Peter so eloquently went through, and all of the relationships and interactions that are taking place at the cellular level, while those of us that are modeling in the field today are doing it at a microscopic level with the bacteria in the rumen and understanding the relationships and the dynamics of feeding that population of organisms to deliver to the small intestine metabolizable protein. And finally, there's the macro system of all the things going on the farm that impact how feed is grown, produced, stored, delivered to the cow in an environment that hopefully is conducive to her optimizing dry matter intake. So we have all those confounders. And so what we have are three different systems that are interacting continuously to hopefully achieve the end result. Now, one of the studies that Peter referenced, that LaPierre did, where they did incremental amounts of crude protein increasing and saw a MP prediction in the model, but no realization of production. Well, that's because in those diets, they were energy limited. And in fact, as the protein went up, they were lowering the energy to keep the package size the same. And so the important part of this is the model as we have it in version 655 does not predict how the cow will partition nutrients. It tells us what is first limiting, whether it's energy or protein. And it doesn't mean that that is what you will get. It tells you what will limit the output of the system, whether it's energy or protein, because the model sees the two interacting dynamically all the time. And so that's a very important consideration when we're reviewing these models and understanding how to use them. And as Peter showed in his latter slides, when we're looking at evaluating what the output of a model is going to be, because we don't know how she will utilize everything in terms of partitioning of amino acids and energy, we can at least look at energy-corrected milk as the summation of all of these effects and use that as the determination of whether we're getting the benefit or the optimization out of the interventions that we're doing. And so I just want to toss it out there. Now, version 7 will be a different model completely, and there we will have feedback loops, a very dynamic system along the lines of what Peter was demonstrating at the cellular level, only we will have it at the rumen level, understanding the interactions that are going on there and all the different microbial systems. So this is exciting because I think these are the three levels that we need to understand as we try to improve sustainability of agriculture by doing a better job of utilizing nutrients most effectively and trying to predict as much as possible how the cow can best use them. And with that, I'll shut up.
Well, Bill, thank you so much. I appreciate always what you have to contribute, and I think our webinars are fantastically better when we get some exchange of ideas between you people who really know what you're talking about. Peter, do you have anything you want to follow up with that, or shall we launch into some questions? What do you think?
I'm fine launching into some questions. I thought Bill had a nice summary there. I agree.
Yes, yeah, he's very good with that. Thank you, Bill.
Welcome.
Paula right now doesn't have any questions yet. Sean, do you have any questions? I do have some from Brazil, so let me know, Sean, if you have questions, and I will, if you'd like to, go ahead and speak.
Hi, Maria.
Hi, Sean.
Hi, sorry I was late, but I just like to say a couple of things. One is we had this webinar in Chinese, let's say 24 hours ago in China. There are about 70 participants, close to 80. So I interpreted that in Chinese, so I played my recording, but I tried my best to follow, to translate what Peter wanted to say. So the feedback is an excellent talk. Really opened some of our nutritionists in China, opened their mind to say, well, they already knew, sometimes supplement methionine works, sometimes it doesn't. So they always struggle, try to figure out why. And after this presentation, really, they said, well, really it helped them a lot. So thank you so much for a great presentation, Peter.
Oh, thank you. Thank you. I really appreciate that.
I thought about to invite you to spend maybe a couple of hours with our audience, but then it's kind of in rush. So I thought maybe next time, but it's excellent presentation. I will share with you a link, your talk in Chinese.
Okay.
I'll let you know.
Okay.
It's great. Come to the question. We had a few questions, about 10 or 15 questions. I grabbed the technical manager of ADCO. So Dr. Peng, he helped me a lot answer those questions. I think one question we did not spend too much time on that is one nutritionist asked about, you mentioned that it's very important to pay attention to leucine, isoleucine, those amino acids, but in current MTS software, what's the best way, what's the best number to check?
That's a great question, and I'll preface it by saying I will give you a recommendation, but I think we need more studies.
Yeah.
I showed you this study, showed you a meta-analysis, showed you a couple other studies. I think we need more studies to come up with a good recommendation like we have for methionine and lysine. Mm-hmm. However, I do think it is important to at least look at it. And so what I do when we're working in the field, first look at total essential amino acids, grams per megacal of ME, often around that 20.5 to 21 target. And then if we would look at leucine and isoleucine, also looking at that relative to ME, and on leucine, I believe, for example, in the Cal study I did, I believe we were at 3.7, 3.6, 3.7 grams per megacal of ME. And isoleucine, I actually sort of forget right now, but I could send you an email related to that. However, definitely put quotations around those in the sense that I think we need more research, and those numbers aren't well defined because it's we simply don't have many studies to draw that from.
Yeah, definitely.
Bill, not to put you on the spot, do you have experience with what you might recommend?
Hi, Bill.
Hey, Sean, how are you?
That's good. I give you a call later. It's very nice to hear your voice.
Thank you. Yeah, the visas are open again, so I just got notice today.
That's great. Okay. Yeah.
Yeah. No, Marianne. So here's Peter alluded to this earlier. Before we even start talking about amino acids, it is so paramount that we make sure we've optimized rumen production of metabolizable protein. To know that we're going to be delivering everything we can possibly, and so often this becomes where the first limiting step is in so many problems on dairies when troubleshooting rations. And it gets back to, unfortunately, not amino acids, but it gets back to the fact that our carbohydrate fractions oftentimes fail us in terms of rumen availability. And again, the model is contingent upon predicting where something is going to digest to optimize the nutrient value to the system. And so often our carbohydrates do not ferment in the rumen to the extent that we need and don't make the bugs that we want and go on to the small intestine where they utilize as energy, but not for metabolizable protein. So my answer is no, I do not have anything I can add to that. But it begs the question, before you start looking at amino acids, make sure that you know you've got the most MP coming out of that rumen fermentation as possible.
Yeah. Yeah.
Agree. And I think in the field, I don't know if you could comment, Bill. I mean, I think it's more than we would expect where that's the most limiting issue.
Absolutely.
Carbohydrate
Absolutely.
fermentation, micro, and having fermentable starch and fermentable NDF as we would expect, and it's not amino acids.
Our ability to characterize proteins adequately for the models far exceeds our ability to adequately characterize the carbohydrate fermentability fractions.
Mm-hmm.
Because starch is not starch. Sugars we're pretty good at, and the fibers, I think we're getting there. That's very evident. But starch drives the rumen. Let's face it, that's the number one. And we really don't have a good handle on that yet. I mean, we're better than a guess, but we have a long ways to go there.
Mm-hmm. And I'll just add, I guess, one other thing while we're talking about that. I think corn silage, especially here in North America, are usually our primary forage. And of the protein in corn silage, I'm a co-author on an abstract at ADSA, and as the starch content changes in a corn silage, the amino acid profile of the protein changes pretty dramatically. Now, you're only going to get 40-50% of that protein bypass, but the one amino acid that changed the most was leucine. So as I had more starch, more kernels, I had much more leucine as a portion of the protein. As I had less starch, leucine was much less because of the plant protein, how it carries it. So that's sort of a side note there, but I think we can start making recommendations, and I'm hesitant to do it myself on, say, how many grams of leucine to feed, because of many of the things that Bill's mentioning there. There's a lot of work to do on that side before we start getting into too many details here.
Question for you on that, Peter. Of those corns that you looked at with the starch levels and the leucine levels, were they flinty corns, or were they softer starches, the amorphous starches, or was it all varieties?
That's my understanding is when we did the meta-analysis, it was using Cumberland Valley data. It was all corn silages.
Okay.
But now you bring that up, that would be very interesting to look at 7-hour starch, even though it's not a perfect proxy, but try to separate out is there differences?
Lindsay, your class is on. Sorry.
What was that?
Sorry.
Oh.
I thought my daughter was her Zoom meeting. Sorry, I should turn off the mic. Sorry about that.
Of flinty versus flowery. So what we did was over all
Yeah. Okay.
And I'd be happy to send you the PDF of it, because the differences in leucine really surprised me across a typical range of starch.
And that leucine is not part of the Zen matrix, right? Zen protein matrix. Or is it?
It was, as starch went up, you got more leucine.
Okay.
So I'm guessing it's part of that, and
Yeah.
it's less. There's less leucine content in the plant protein.
Got it. Okay. Yeah, I'd love that. I'll get you my email address.
Okay.
Thank you.
Yep.
Yeah, thank you. Thank you, Sean. If you have more questions, let me know. I'll get started on some questions for Sean. Do you have more questions?
No, thank you. My daughter's
Okay.
long. Yeah,
Right.
sorry.
Yeah, thank you for all the effort you put into making sure that it gets delivered as well to your Chinese audience. That's greatly appreciated.
Okay, thank you, guys.
Thank you.
I will start in on some questions that I have from Marcos. So his first one is, it's very interesting, the data on nitrate level of counties in Idaho. Are there visual or measurable signs of health issues in humans or other animals directly related to the high nitrate levels in this region?
Good question, and I don't know the answer. I guess I can send maybe you, Marianne, where I got that information.
Yeah, that'd be good.
The researchers, Dr. April Latham, she does a lot of research in protein and nitrogen related to animal livestock production. She's based in Idaho. And I can also send a link to hers, and maybe she could help answer that question. What I was showing with just the major nitrates, the effects of that to the human population, I don't have an answer. But I'll work on sending that to you, Marianne.
Yeah, and if you don't, if it's okay with you, some of the other links that you've referenced, if you put those together sort of in a little bibliography, if you don't mind, I'll send it out with a link when I have the recording completed.
Okay.
That would be great. That way people who are listening can also benefit from those. I see that Paula is starting to get some questions. I'll do a couple more of Marcos, and then we'll go to some questions from Argentina. So the next one is in relation to some soils. Do you know the depth of the water, how the depth of the water source affects the chance of contamination of groundwater with nitrogen? I assume sandy soils will increase the chance of contaminations relative to clay type soils.
Again, I think my assumptions in line with that. Maybe I'll defer to Bill on these questions. But I provided it sort of as a background on a reason for wanting to study this, but I don't have answers related to that.
Yeah, so my understanding, and it's rudimentary, percolation is the big thing, how quickly it goes down through, and then if there's any type of epiphytes, the organisms that are in the soil that would capture the nitrogen and break it down, whether it's vegetation or whatever. But I'm not sure, having lived in Idaho, I probably should know the answer to that, but I claim ignorance there. It wasn't an issue where I lived. But the soils in Idaho tend to be fairly compact, and they suffer from erosion because they dry out on the surface, but it's tough to dig. So penetrating that, I think, is a challenge. There's more runoff that I recall in heavy rains than there is actually saturation of the ground. But I would defer that to an environmentalist, and I'm sure the soil type in the region is going to vary and such. So I don't have a good answer. That's my answer.
I wonder if we might have some information on that on the webinar that Frank Mittler did and Jude Tapper last month. Frank was in April, and I know we did cover some of that a little bit. One of the things that, oh my gosh, Carl Zemek, who's with does a lot with environmental planning at Cornell near us, says that a real issue that can be in in at least New York State and other parts of the US where they've done a lot of tiling. It does change just even the natural response to that the soil would have to some of the percolations. And so you get some really fast movement through the soil of fluids that you wouldn't naturally get. And that leads to some issues with environmental concerns.
Yeah.
But I'm going to do one more, and then we'll let Paula have a turn talking. What would be reasonable goals for the marginal efficiency of converting MP and digestible methionine into milk protein? For example, grams of milk protein per gram increase in metabolizable protein, or grams methionine above a basal diet to achieve a reasonable income over cost. Is it possible to even have a number, or is that kind of dependent upon the situation?
Excellent question. If you're just looking at milk protein, I think we can make an elementary calculation there. And if you're looking at just metabolizable protein, I would look at the price that you're paid for milk protein. So what is my revenue if I change milk protein? And then I would look at the cost of MP, and that, of course, as we know, that varies. So the price of MP, which, for example, you use soybean meal, that's going to vary, and then the price of milk protein is going to vary. When in normal situations, and I've done some analysis looking at this, that's going to vary. But in normal situations, if I can get 20 to 25% marginal efficiency, so if I add 100 grams of MP and I get 25 grams of milk protein, it's usually favorable economics. But again, I'd encourage you to do the calculation on that given farm. But I think 20 to 25%, 25%, 30% being on the high end. If you're getting responses less than 20%, say 10%, I would evaluate the economics. I'd evaluate energy. Definitely looking at energy when I'm making these changes. But on the high end of just looking in literature, it's 25 to 30%. Caveat is fresh cows. If you're looking in fresh cows, say day of calving to 40, 50 days of milk, you may get 40, 50, 60% marginal efficiency. Post peak, I would try to target that 25%. Methionine, looking at that. So again, if you're driving up energy at the same time, I'm providing a good amount of other essential amino acids, rumen fermentation's optimized, I think getting 20% is reasonable. At times, there's studies out there showing 30%. And this is a calculation you can do on your farm. So if I add 12 grams of metabolizable methionine, how much does milk protein yield change? And what's the concentration of methionine in milk protein, which is usually, if you account for the casein and whey, it's around 3%. So if you do that calculation, I would encourage you to do that when you do make a change. If you're getting 20% or greater, I think you're making the right, you're doing a very good job of amino acid balancing. If you're getting 10% or less, I would question, is there an issue on your energy, on your rumen fermentation, on other essential amino acids there as well. And of course, again, when you're looking at methionine, look at the economics. What's the price of milk protein? What is the cost to supplement the methionine? So I think from the question is excellent, and I think the calculation can be made. Though there is uncertainty around the calculation, just looking at milk protein, but that calculation does not take into effect milk fat, if there's a change in milk fat, and we know there can be, and also health, other benefits that come from amino acids. But excellent question.
Thank you. I have a couple more questions from Marcos, but we're going to switch over to some questions from Argentina. Paul, this morning, I know we talked a little bit about MUNs and as a measure of efficiencies. And Paula, I think, has some questions regarding that. And also a really good point on some of the other things that aren't milk that maybe we need to consider protein for. Paula, go ahead.
Hi, Peter. Nice to meet you.
Nice to meet you.
Okay, I have a more general question and not specific about amino acids, but can you make some comments about considering MUN as an indicator of global amino acid balance in the cow, playing with changes in fermentable carbohydrates, and also considering milk protein? I mean, watching the response of a change in fermentable carbohydrates on both MUN and milk protein response.
Yes, and we sort of touched on this morning. I mean, I think the first point, and I think, I believe Bill made this point, lab accuracy of MUNs is a caveat that should be noted. But if you're confident in your MUN results, looking at carbohydrate, rumen fermentation, and the protein that I'm supplying to the rumen, optimizing that appropriately, MUNs can be very influential looking at that. The other caveat I would add, I think sometimes we forget that overfeeding MP, overfeeding bypass protein, can also drive up MUNs. So MUNs, I think it is an excellent proxy for saying, am I optimizing rumen protein efficiency appropriately? If I have an MUN above 12, say 14, 15, do I have a lack of fermentable carbohydrates, or am I overfeeding degradable protein? Caveat being in the case that we're not overfeeding bypass or MP protein. On the low side, I think when you approach 5, 6, or 7, especially on a herd level, are we causing some issues with rumen fermentation there, either too fermentable carbohydrates or not enough protein. So I guess that's sort of how I look at MUNs. I don't, I'd look at it, but I don't, I guess, put too much emphasis on it. I think looking at an individual herds and seeing how it changes from week to week, and what the trends are, and when there's new forages or a change in ration formulation, how does that change? I think is very important feedback loop to you as a nutritionist, how that changes being implemented in the cows. I think you had a few questions about like just milk protein. I'd encourage you to look at that. I mean, if you have good intake and you know milk protein and you know the crude protein of your diet, you can calculate nitrogen efficiency. And I think AMTS predicts that or does that. So I think it's a good metric to look at. Am I at 25% nitrogen efficiency gross, or am I at 30%? I think if you can get to 30%, that's a great accomplishment. If you get to 35%, you're well above average. So that's another way I would be looking at efficiency is just what is your overall nitrogen efficiency on that group of lactating cows.
Okay,
Paula, while Peter just brought that up, I do have a question that relates to that. So if you don't mind, I'll jump in. And then I think the next question you're going to ask, I have a similar one too, so maybe we can do it jointly. So this is from Marcos. They have performed some nitrogen balance experiments with lactating cows for the last 3 years. Gross nitrogen efficiency for milk is usually between 22 and 29%, and usually higher on lower protein diets than on the high protein diets, as you would expect. What interests Marcos is the variation in what they call nitrogen retention. Nitrogen in the urine and nitrogen feces is never equal to nitrogen intake. So retention equals the intake minus the urine and feces nitrogen. Retention gets the errors of all the measurements, but experimental methodology is as consistent as possible. The mean nitrogen retention of 4 experiments involving 14 treatments was 12.7% of nitrogen. Experiment means were 3.8, 11.4, 17, and 18.7. Could you comment on this? The values can be high compared to the nitrogen loss in urines and feces. What is the biological meaning of this, and can it be manipulated, and to what degree? Peter, do you want that question put in the chat window so you can see those numbers?
Yes, I think that would maybe be good, and I don't know, could I respond by email, possibly a later date too? I guess my first question back to is this: are all studies primiparous or multiparous, or is there a difference in the type of animals being used?
Marcos, do you want to comment on that, or what do you know? Both primiparous and multiparous.
Okay, across all the studies, it's not like one study is primiparous and one study multiparous?
It was blocked by parity.
Okay. Then I don't have a good answer for you. I mean, the one thing I think is really strong, and a recent experiment I saw at Ohio State, in primiparous cows, first lactation, there is significant growth and need for protein, retained protein for growth in those animals. So in multiparous, that's going to change as you approach maturity. So some of that variation you're seeing of retained protein for growth, I think is a reflection of animals that are growing versus animals that are not. Nevertheless, that is a lot of variation, and does that reflect, I guess, this concept of, is there certain animals that are different in efficiency? I think at some point, the whole residual feed intake, do we start looking at that residual nitrogen intake and start to think, is there a genetic relationship to certain animals versus other animals? And there's probably definitely a possibility there as well. I guess on the measurements, I mean, were the urinary nitrogen, was it measured, or was it predicted? I guess would be some questions I have, and how that relates, but I can respond offline too, unless we want to continue this discussion.
It was a total collection of urine and feces, and Marcos too was amazed at the measurement, the variation within them.
Okay. Were they able to like age? I know it's maybe not most statistically appropriate, but maybe just looking at age or body weight or average daily gain, or did they do any muscle measurements or anything related to looking at how much growth is going on, or no?
I think you and he probably at this point can correspond back and forth. I'll share the connection, the email with Marcos, but he thanks you for considering it. He'll email you. Interesting when you said the potential genetic variation. What about epigenetics? How that's like another variable.
Agree, and I think, I mean, there's both ways. We have very good evidence on methionine as you change methionine status and DNA methylation and the imprinting that could have on the next generation. And so I think we see that certain amino acids can affect metabolism of the next generation. And then also, I guess, of that next generation, do they have differences in protein metabolism? And I don't know if that's been looked at. So if you have an animal that's been altered from a nutritional intervention with epigenetics, what is the outcome on that? Is there a change in protein metabolism or not? And I don't know.
Paula, back to you. And when you ask your question about the different amino acids, I'll chime in too.
Okay, this is about methionine supplementation under certain circumstances like heat stress, different lactation periods of the cow, like a fresh cow.
You just like some comments about methionine in those circumstances?
Yes, specific supplementations for those cases.
I'm not too experienced on the heat stress, so I'm going to comment first on fresh cows, transition cows. Yes, I think there's several studies, some studies doing pre-fresh and post-fresh and seeing responses. Some studies doing only post-fresh and observing responses. And I think some of that is due to just simply a substrate requirement. You have low intake and you're trying to produce milk and early lactation, so negative protein balance. Definitely a need for methionine there, and it probably is prioritized towards the mammary gland, which it actually is prioritized there. So the methionine available for other tissues, liver, other tissues throughout the body, as the other functions on inflammation, etc., methyl donors, definitely some benefits there where we see a lot of, I guess, indices related to health improved. So benefits from methionine definitely there in fresh cows, and I think some of it is the mammary glands taking up most of the methionine, and you're in a deficit, and so methionine available for other uses is really limited. Related to heat stress, I haven't looked at much research there, so I don't have a good answer for you. I will say I'll speculate, I'll hypothesize. Intake's reduced, and so I think some of the same situation can occur, though you are having corresponding reduced milk production substantially. But from a health immunity standpoint, there can be some benefit. What I'm also talking about this, I will say I think sometimes just focusing on methionine, is there a point where we could be overfeeding methionine? There is some research or literature out there showing that feeding too much methionine decreases blood flow. In humans, overconsumption of methionine leads to insulin resistance. So I think we need to be a little bit careful in that heat stress situation with insulin dynamics, et cetera. If I feed too much methionine, how is that all interacting? And I don't have a good answer on that, so I'm just sort of speculating here because I've not read much literature on that subject.
We have a couple of webinars coming up. Jennifer Van Oss is going to talk about heat stress in August, and then Marty Traxler is actually our speaker in November, and perhaps he'll be addressing some of those things. I'm not sure if Jennifer's is going to be focused more on sort of the management aspects of it rather than the nutrition. So that we may be able to have that, and Marty's with us tonight if he felt like adding any comments, he could type it in. But go ahead, Paula.
Okay, and the last question is from Abner. Should we consider leucine and isoleucine as most important amino acids compared to arginine? Which would be the recommended values?
So just so I understand the question, you're saying, should I consider leucine and isoleucine as more important than arginine? Is that the question?
Yes.
Okay. My first answer is I don't know. So what I would say is, again, my study just looked at leucine and isoleucine, and we did not look at arginine. There are some studies out there showing responses to arginine. You look across literature and other species, arginine, there is a mechanism for direct connection to mTOR, so stimulation there, but it's not as sensitive as say leucine. So intracellular, so the concentration of leucine in a cell, that is more sensitive to mTOR at a lower level. To get stimulation from arginine, you need a much higher level. So from a mechanism standpoint, I would say leucine. Now that's looking across multiple species. In dairy cows, I don't know. There are some studies looking at it, but I just don't know. And I'm hesitant, very hesitant, especially to say that leucine is more important in dairy cows, because I don't think we have research to directly back that up.
Just to follow up with, Marty says that he's going to focus primarily on management of heat stress and abatement methods that they use because he's down in Mexico. But he thought he would maybe look into adding some nutritional aspects as well. So thank you, Marty, for that response. While you're talking about the separate amino acids, I had a question from Marcos that was, how important is histidine to amino acid balancing? How would you consider the potential of histidine supplementation to elicit milk protein response relative to leucine or isoleucine? And why valine has not been a stimulatory type amino acid? And I'm sorry if you answered some, I was maybe not paying as good attention as I should have.
Yeah, no, this is I really like the question, because I have the same question as well at times. But histidine, I would say, I view it more as more like a requirement type amino acid. And I think, especially when we're feeding lower protein diets and relying more on microbial protein, I believe a histidine becomes deficient. And there's several studies to support that. So when I'm looking at formulating diets, I usually like to have histidine at least 1-to-1 with methionine, preference target of 1.1-to-1 of methionine. So if I'm looking at it relative to energy, that would put me at 1.2 to 1.25 grams of histidine to megacal ME. Looking at from a stimulation standpoint, you look in literature across other species, and you don't see that direct mechanism to mTOR. Cell culture work in mammary, like dairy cow epithelial cells, not near as significant of a response stimulatory as leucine. So just broadly categorizing it, I would categorize histidine more as a limiting amino acid, not a stimulatory. And I think, especially when you get in a lower protein diet situation, I would be concerned, and look at it relative to methionine is how I look at it. Regarding valine, I think that's a very interesting amino acid, in the sense that a lot of studies will talk about branched chain amino acids and say branched chain amino acids are limiting, so let's do an experiment and let's infuse a lot of branched chain amino acids. Well, valine has not been shown across other species to have a direct link to mTOR. There's, from a mechanistic standpoint, it has not been found. Not saying it won't be found, but there's no evidence yet to support a direct link to mTOR. So I think using it as a stimulatory amino acid in conjunction with leucine, I don't think there's a benefit. Actually, I think there's possibly a negative effect, and that relates to transport. So the branched chain amino acids, valine, leucine, use the same transport systems to get inside the cell. And there is some evidence out there, if you have too much valine, or it can compete with leucine to get into the cell. So if I'm increasing branch chains all at once, or I'm driving up valine, I may decrease the ability of leucine to get into the cell, thereby negatively affecting milk protein. And from a high level, to sort of further support that hypothesis, Dr. Mark Hanigan, there's been several abstracts on it. I don't think the paper's been published yet, but across very large data set, and I think this is going to be included in NRC. Valine had a negative effect on milk protein. So as I had higher valine supplies, higher metabolizable valine, milk protein went down. So I think that's part of the reason when I did my experiment is I don't want to think just about branched chain amino acids. I want to think about, from a mechanism standpoint, how do they relate? And I think my hypothesis at this point is valine is actually a negative on milk protein. So sorry for the long answer, but I hope those questions.
Thank you, Peter. Paula, do you have any more questions? I am out of questions. Unless somebody writes one in, I
don't.
Okay. Bill or Sean, any questions, comments?
I think that the relationship between so many nutrients that seem to be confounded gets back, I think, to the point that when we satisfy what is first limiting, automatically then something else now becomes first limiting because we've satisfied what the immediate barrier to production or progress was. And I think understanding those interactions is where the devil's in the details. And as Peter pointed out, there's just a lot of these things, whether it's competition for a transport mechanism or whatever. And I remember Sniffen and Chalupa telling me over and over, too much methionine, you'll lose milk. I didn't know why, but I took that to the bank. And so there's a lot of these things that as we pursue these things at the molecular level, we're going to have a greater understanding of how we can modify at the macro level to prevent these type of conflicts. So it's but more questions than answers right now, I guess.
Thank you, Bill. And Sean says he has no more questions. Paula has no more questions. Everybody, thank you for attending. And I think since we don't have more questions, we can plan on meeting here next month for talks about heat stress. And Peter, thank you so much. And Bill, I special thank you to you. You did this twice today. That's above and beyond, and I really appreciate it.
When the complaints come forward, you'll recant that. But
nobody ever complains.
Send me Peter's email address so I can connect with him. Okay.
Yes, I will do that.
And thank you, Bill, very much as well.
No, thank you, Peter. It was good. I look forward to connecting with you in person.
Yep. And thanks, Marianne.
Thank you, Sean. Thank you, Paula. Everybody.
Here, Sean.
Yep. Look for it. Look for the recording in a week or so. And everybody else, thank you and have a good night.
All right. Thank you.
Yep.
Bye.
All right.